Hello students, we are again back on the platform of biology and ethics with the lecture number 2 of embryology. We have already studied the lecture number 1 of embryology, in which we have studied the formation of blastocyst from the zygote stage. It means zygote has changed into the morula, morula changed into the blastula, blastula into blastocyst, and further there was a hatching of blastocyst. Now further, after the hatching of blastocyst, what happens? So, beta, we know after the consign of zona pellucida was removed and ruptured, the inner cell mass and outer cell mass, which was trophoblast cell, has came out from that just like the hen, just like the hen hatches outside from the egg. That same similarly, the blastocyst has been hatched out from the consign of zona pellucida. Now, after coming out from the consign of zona pellucida, we know that the zona pellucida was inhibiting the ectopic pregnancy in the female's body. Now there is no zona pellucida, then there will be implantation. See here. This is the wall of endometrium. This was the outer cell mass, which was called trophoblast cell. This trophoblast cell start entering inside the wall of endometrium, and there was an inner cell mass. This was the inner cell mass material, and this was the blastocele. This was the blastocele cavity. Now, further, these trophoblasts comes further in the division and start increasing their number inside the walls of endometrium and makes their connection strong. Further, these cells convert them into the chorion, the extra embryonic layer. After some time, these cells differentiate themselves into a structure like finger-like projection like this. This is chorion. This is beta chorion. This is beta chorion. And chorion is an extra embryonic layer. Extra embryonic layer. And we know that the outer cell mass, it means I am talking about the trophoblast cell, contributes in the formation of placenta. We have already read in the lecture number 1 of embryology. I am repeating this beta. It is very much important that the outer cell mass, the trophoblast cell, contributes in the formation of human placenta, and inner cell mass was contributing into the development of embryo. Now, what happens? These are chorion, and the finger-like projections are called chorionic villi. These are the chorionic villi. These are the chorionic villi. And I want to tell you bit about this chorionic villi. This chorionic villi are capable of determining the gender of a fetus in the amniocentesis process. It means if a parent or a couple is going for determining the gender of a baby, the sample of chorionic villi is also capable for determining that. Okay. Now further, after the formation of chorion, the extraembryonic membrane or an extraembryonic layer, there is also one more extraembryonic layer which is called allantois. This was the first extraembryonic layer. Second extraembryonic layer is allantois. And by the help of these two extraembryonic layers, together they will form placenta. There will be formation of placenta by the help of two extraembryonic layers, which are allantois and chorion. That's why human placenta beta is also known as chorioallantoic. Human placenta is also known as chorioallantoic because the human placenta is formed by the help of two extraembryonic layers, which are chorion and allantois. That's why it is also known as chorioallantoic. Now, these placenta releases 4 hormones of itself. There are 4 hormones of itself, which are HCG, which is human chorionic gonadotropin. Second, HPL. Third, progesterone. And this progesterone was released by the placenta in a very large amount because the excess amount of progesterone leads to the inhibition of myometrial contraction in the female's body. And then fourth one is estrogen, and this is released in small amount by human placenta. Okay, the thing to be reminded specifically is that the placenta is releasing progesterone in a large amount because progesterone large amount will inhibit the myometrium contraction better. Because we know that when there is an implantation on the walls of endometrium of blastocyst, there will we have to inhibit the myometrium contraction for the safe pregnancy till the gestation period. So the progesterone is released by human placenta in large amount, which will inhibit the myometrium contraction in female's body, and estrogen in less amount. But what happens when a fetus is developed by a virtue of time? When a fetus is fully developed, then the signal will come to the human placenta that development of fetus has been matured. Then the placenta will reverse the secretion. It means now after the development of fetus, progesterone will be released by placenta in small amount and estrogen will be released by placenta in large amount, which will lead to the contraction of myometrium wall in the female's body. And if there will be a myometrial contraction in female body, it will lead to a labor pain on the mother's body. That labor pain will affect the hypothalamus. Hypothalamus will send the message to the posterior pituitary to release more and more oxytocin. That was also known as birth hormone. And that oxytocin was again acting on a myometrium for more and more contraction. By the virtue of increasing more and more labor pain in the mother's body, the labor pain will lead to the increase in stability, capability of mother's endometrium for more and more pain. After the more pain will arrive, that will again send a sequence to the hypothalamus, and again hypothalamus will send a message to the posterior pituitary to release oxytocin. This is an autonomic activity. It means it is a reflex action. We have read in the fetal ejection reflex. This was a reflex action which finally leads to the parturition in mother's body. But better, we have already read that the outer trophoblast cell was making the chorion and having a part. But what about the inner cell mass? Now, in this video, I am focusing on about what will happen by the inner cell mass. But first, I want to say and give you explanation on SCG more, because there are more probability of coming questions of SCG in NEET. That basically, beta, these SCG was released by human placenta, was going in the mother's body in the ovary to maintain the corpus luteum for a safe pregnancy. But after this act, mother's body eliminates that human chorionic gonadotropin in the urine. It means mother is eliminating that SCG after the action in the form of urine. That's why the sample of mother's urine is used for the determination of pregnancy is being established or not. It means a doctor uses the sample of mother's urine to test the presence of human chorionic gonadotropin in that. Because when the fetal cell will release through by placenta human chorionic gonadotropin, these human chorionic gonadotropin will go to the mother's body in the mother's ovary and maintain the corpus luteum for the safe pregnancy. And if there will be SCG in the sample of mother's urine, it is very much saying and proving that there has been an established pregnancy. Okay, so this is used for the detection of pregnancy in the mother's body. Now, after this, we will talk about what about the inner cell mass. We have already read about the outer cell mass, what was happening by the trophoblast cell. But what about the inner cell mass? This inner cell mass goes and contributes in the development of germ cells. It means I am talking about the gastrulation. I am talking about the gastrulation. It means the inner cell mass was contributing in gastrulation. In other words, the inner cell mass was contributing in the development of 3 germ layers, which was, I am going to show in the diagram. You can see here the diagram, which is very much visible that the inner cell mass is being divided into 2 categories of cell. One are longitudinal cells and some are spherical. And bilayer, this is a bilayer blastocyst. Now, the upper are called epiblast, lower was called hypoblast. These hypoblast is making a cavity. You can see here, making a cavity. This cavity is called yolk sac, and these yolk sac are absent in humans. Or we can say these are temporary. These are the temporary cavity in humans. And further, you can see here, beta, these hypoblast and epiblast cells. We'll see like here, beta. These are the trophoblast. These are the outer cell mass, beta, which are called the trophoblast. The inner cell mass was divided into two, the hypoblast and the epiblast. These longitudinal cells are epiblast and these spherical cells are hypoblast. And this cavity is called yolk sac, which is absent basically temporarily in human being. By the virtue of time, this will disappear after some time. Okay. Now what happens? These epiblast cells, these are, I am writing here, epiblast, and down this was hypoblast. These hypoblast cells has further continued and captured the area and formed a cavity. This cavity is being captured and formed by these hypoblast cells. And these hypoblast cells does not contribute in an embryonic development. These will not contribute in the development of an embryo. Only the development of embryo will be on the hands of these epiblast cells. Now further, after some time, these epiblast, above the epiblast, there will be a cavity, formation of startup of a cavity like this beta. These trophoblast cells will increase and form a cavity here. Here was the epiblast and here was a cavity. This cavity is called amniotic cavity. This cavity is called amniotic cavity. And the cells which has formed this cavity are called amnion. The amnion. Now you have understood that what is the amnion. This word amnion has came very much frequently in the embryology. This was the amnion. We are talking about this amnion. These amnion are forming the cavity above to the epiblast cell, which was called amniotic cavity, and the fluid which is present in this cavity is called amniotic fluid, and this amniotic fluid is used in the process of amniocentesis for the determination of the sex of a baby. Okay, the amniotic fluid is also capable. Behind we have seen the chorionic villi was also capable. The sample of chorionic villi was also capable for the determination of sex in the baby during the amniocentesis, and the amniotic fluid which is present in the amniotic cavity is also capable for the determination of sex in a female body or a baby. Okay. Now, these middle portion, which it means I am talking about this epiblast cell. This is called beta. This is an embryonic disc. This is embryonic disc, and this embryonic disc will further go for the development of three germ layers. Three germ layers, which are ectoderm, mesoderm, and last the endoderm. These are the three germ layers which was being formed by this embryonic disc, and the process of formation of the three germ layers are called gastrulation. Gastrulation. So this is gastrulation, the formation of three germ layers. And after the formation of these three germ layers, there will be lead to an organogenesis. It means there will be development of slowly development of organs. We will differentiate some. Slowly and with the virtue of time, organs will be developed. Some organs will be developed from the ectoderm. We will say the ectodermal origin. Some from the mesoderm germ layer. We will say the mesodermal origin, and some from the endoderm germ layer, which we will call the organ is developed from the endodermal origin. So the development of these three germ layers is basically termed as gastrulation. So we have come from a topic gastrulation. Now after gastrulation there will be lead to an organogenesis, and organo means the organ, and genesis means formation. There is formation of organs from these three germ layers. Some from this and this. This was the concept till the development of organ. You can see better here the chart given that at which month, at which event, which organ is starting for the development. From very first, the human heart, the baby's heart gets developed from the first month. You can see this chart. This chart is for y'all, and this chart should be in your mind because many of the questions of PYQs from this topic is being asked from this specific chart. Okay, so take a screenshot of that, or you can note in your copy and any other else. Okay, now being continued in my channel because on my next lecture we are going to discuss further. Thank you.